Hello everyone, thank you very much for visiting our poster logic-based classification of macrophages. Myeloid and lymphoid lineages derive from differentiation of hematopoietic stem cells in the bone marrow, where common myeloid progenitors further differentiate into mature cell types like monocyte and terminally differentiated cells such as macrophages. Although there is a wide variety of macrophages in vivo, both M1 and M2 macrophages have two distinct functional roles in vitro. Whereas M2 macrophages shown to promote the tumor growth, M1 macrophages represent anti-tumor properties. Therefore, it is necessary to formulate a monocyte macrophage cell lineage to control the tumor prognosis, as shown here. Since each blood cell lineage is produced in different body conditions, but the necessity to have the macrophages in long time causes the transformation of macrophages which help in tumor growth. Consequently, the need for an increasing number of macrophages, the number of cells in the same lineage increases. Therefore, the linear relationship between the number of cells required to be removed to avoid producing M2 macrophages. In other words, the biological process needs to be changed in a cycling form from hematopoietic stem cell until monocyte whenever there is a need to have macrophages in the long run. To avoid tumor growth to reach to a metastatic state, a rotating management model comprising the brain, gut, and spinal cord is suggested, where the alternative drug targets to prevent inflammatory conditions are introduced in 7 categories based on the importance of the topological positions of the network. Here, a rotating management system is introduced to avoid metastasis. To check the feasibility of drugs to address the emerging issues caused by cancer, again with a few rules is defined, comprising of 5 stages of cancer and 5 categories of drugs. And it is highlighted that death can be prevented as long as cancer does not reach to a metastatic stage through changing the management roles in the management system. The management system here includes brain, gut, and spinal cord. A minimum requirement to guarantee the feasibility of the drug treatment for each level of cancers are shown by arrows. To introduce 7 categories of the drug targets, we relied on our well-developed methods in the past, such as identification of key player genes in gene regulatory networks, where we introduce 2 sets of dominators and in the minimum dominating set solution and dominators and connectors in the set of minimum connected dominating set solution. We also relied on the method that we introduced in the paper, identification of biomarkers controlling cell fate in blood cell development, where we introduced several biomarkers such as cell fate biomarkers, influencers, and connectors. Since some of the genes in the particular cellular identity play roles such as master regulators, here the highest color in the color spectrum, red, assigned to them, and cell fate biomarkers, dominators, hubs, connectors, influencers, and targets with orange, yellow, green, blue, cyan, and purple respectively. And the main goal is to consider the healthy genes in each category to fulfill the dysfunctioning of the mutated ones in the same category in the frame of drug repurposing. This idea stems from our tool, Top Control, which is a tool to prioritize candidate disease-associated genes based on topological network features, where in this tool, at first we consider all the genes in the first level, then differentially expressed genes in the second layer, and the differentially expressed genes which construct a network in the third layer, and the dominators, hubs, connectors at the top layers. Thank you very much for your attention.